Clotting factors were first identified as those which were lacking in individuals with a particular type of haemophilia. Factor VIII used to be, sometimes still is, called anti-haemophilic factor because it's something in blood which was shown to correct the clotting time elongation that you find in blood from a patient with haemophilia. That was first established in the late 19th century by Almroth Wright, the mentor of Sir Alexander Fleming, the bacteriologist who discovered penicillin. But historically, Almroth first showed that blood from a haemophiliac was slow to clot in a glass tube. Moving on from there, during the last century, the individual factors were gradually processed to the point of being demonstrable as biochemical entities, but putting their function together in a cascade was the work of the most influential British scientist in this area, whose name must be mentioned, McFarland. And McFarland worked at Oxford, and his interest was in haemophilia. He set up the first haemophilia centre, and he also set up the processing of factor VIII from slaughterhouse blood from pigs and cattle. And until fairly recently, we were using a version of factor VIII extracted from pig blood to treat patients with resistance to the human form of the factor. Now, my interest was really sparked in the process of the purification of the individual factors, because that was the key to the next advance, hence my devotion to factor VIII, the last of the circulating clotting factors to be purified by ourselves. But there was another factor that I've been involved with, its purification and cloning, and that is tissue factor. Now, that's not circulating in the blood with the other clotting factors. It sits waiting around blood vessels and in tissues, waiting for that moment when it's called into action by a damage that releases blood into the tissues. And in blood, all the clotting factors circulate in inactive form, soluble, but as soon as blood spills into tissue, it contacts tissue factor, which is not in the circulation. It exists as a transmembrane protein, which binds tightly to the circulating factor VII. The complex of tissue factor with factor VII is then activated. Factor VII on its own has no activity, but it then activates factor IX, which can then activate factor X, along with the cofactor factor VIII, which in the circulation is in an inactive precursor form, bound quite tightly to its carrier molecule, von Willebrand factor. That interaction, by the way, has continued to be a major interest of mine, and we, with our helpers in the biophysics department, solved the structure of the module on factor VIII that is bound to a module on von Willebrand factor. Now, all of the factors are proteins. It's the sequence in which they interact which gives rise to a biochemical amplification cascade. And the first scientist, as I mentioned, to realize what was going on was McFarland with his Cascade hypothesis, simultaneously with workers in the States, McFarland and Ratnoff and Davy, correction, who published their waterfall hypothesis that was essentially the same hypothesis. So we can go from an initial trigger of a minute amount of tissue factor, probably in the picomolar range, with a minute amount of factor VII that then converts a small amount of factor IX, that with factor VIII converts a larger amount of factor X to, with factor V, convert a much larger amount of prothrombin to thrombin that then converts fibrinogen to fibrin that spontaneously polymerizes to form the solid clot. The amplification in that cascade over a minute is at least 1,000,000-fold. So as soon as you start to bleed, that process gets going, and within a minute or so, there's 1,000,000 times as much thrombin converting fibrinogen to fibrin, the clot forms, and the bleeding stops.